Good morning. First of all, I would like to thank Doctors Sarah Felstein-Ewing and Dr. Tammy Chang, not only for organizing this symposium, which is of tremendous interest to NIAAA, but also for inviting Dr. Huebner and myself to give some joint introductory remarks. We both are very interested in this conference because it brings together different areas of research, namely brain imaging, which is in my division, and treatment and research, which is in Bob's division. And we think that at NIAAA, moving forwards, we're very interested in a translational research approach with the ultimate goal of improving treatments for alcohol use disorders and alcohol dependence. Now, first I'd like to say a few words on the neuropsychology of alcoholism, which resides in my particular division. There have been several systematic neuropsychological studies involving groups of men and women in recovery from alcohol dependence. And these studies have revealed a very typical profile of functional impairments, which includes difficulties with executive function, visual spatial abilities, as well as gait and balance. So with extended sobriety, these impairments lessen and some functions appear to return to normal. However, parallel with these changes, performance changes, they're very well established brain structural changes that to some extent are reversible depending upon the condition. And we're very interested in that aspect. So typically some alcoholics, very much like stroke patients, carry the potential for restitution of function. And unlike hemorrhagic stroke and other head injury victims who incur very definitive neuronal cell damage, alcoholics typically endure what we call as an incomplete lesion. Therefore, they retain neural tissues that could have the potential for structural repair and thereby enable functional repair. I think this is a very exciting thing to distinguish between the kinds of lesions that you see through imaging studies on alcohol-dependent individuals. Now, this potential, I think, is further enhanced by recognizing that alcoholism-related neural damage affects microstructural integrities. And I know that Sarah Jones, who's here in the audience, is going to speak about that. And they can remodel with extended sobriety. So that's the promising end of things. I would also like to make some comments on neural circuits that are affected in alcoholism, because in our division, we're very interested in circuitries. In recent years, when we consider brain insult, we seldom think of it as an isolated event. But now consider it in the context of circuitry that is affected. The bad news about circuitry is that damage in one area can affect distal regions and functions. The good news about circuitry is that healthy nodes and systems intersecting with the ones that are affected have connections of their own and have the potential of taking over affected function or at least compensating for dysfunction. I think this is a very important point. Neural systems known to be affected in alcohol dependence include the frontal striatal reward circuitry, the frontocerebellar circuitry, and the limbic circuitry. And so therefore, there is great potential for targets, especially in terms of retraining the circuits. Functional imaging studies have already shown that for alcoholics to perform certain tasks, such as those involving spatial working memory, at normal levels, they activate the frontal and the cerebellar regions of the brain to accomplish working memory tasks, that controlled subjects can do by only activating the frontal regions alone. Another interesting potential source for functional enhancement is the coupling of task-activated networks with resting state activity, and George Fine has some interesting papers in this particular area. So perhaps by enhancing resting state activity, somehow the brain selects systems appropriate for the tasks ahead, affording another, I think, important source of functional compensation. So in terms of assessment of circuitry repair, the combined use of transcranial magnetic stimulation, or TMS, as well as transcranial direct current stimulation, which is also called as TDCS, is now being considered as a method to redirect selective regions of brain response to very particular tasks or stimuli. And there was a very nice review by Page et al. This year in 2015, and I'm sure all of you all are aware of it, but if you've not read it, I strongly recommend you reading this review article. In addition, multimodal imaging using diffusion tensor imaging to determine the health of the white matter microstructure, together with functional MRI for brain activation, could assist in identifying brain networks that are viable and those that are not viable, and potentially support retraining. And an article in Neurology this year by Arneman scores the point. It's a very good article. I also recommend that to you guys if you've not yet read it. So I think the hope for the future is in fact in neuroplasticity. The idea of retraining the brain and its circuitry to overcome dysfunction is a hopeful concept, and I know it's easier said than done, but it's having success in a number of areas, including stroke, reading disability, dyslexia, and traumatic brain injury. The dynamic course of alcoholism is marked by neural circuitry injury and associated cognitive and motor compromise that may support the reversal of damage or escape of function too. So in conclusion, I believe that a good tool such as brain imaging will support the development of more effective behavioral therapies for alcohol use disorder and will help us understand the brain-based mechanisms of why some treatments work and why some do not. And also why treatments work in some individuals and not in others. So I'm trying to come towards the idea of personalized medicine. Imaging could also be used in the actual treatment process with circuit retraining and neurofeedback during functional imaging as an example, and I think some of you all are doing it. Lastly, the topic of this symposium helps to bridge across our different organizational divisions at NIAAA. In this case, the Division of Neuroscience and Behavior and the Division of Treatment and Recovery Research. It is rewarding to know that despite our different scientific approaches and the areas of expertise, that we can actually work together to promote the mission of NIAAA and support our grantees such as y'all who do such important work, even in very difficult budget times as we are facing right now. So a big thank you to you, Sarah, and to Tammy for organizing this conference, and a thank you to all the speakers and all the participants in this symposium. Thank you.